from Martin Sarter's lab, and I think I've asked Martin to introduce Cassie and give us just a couple sentences about her background.
All right, Cassie, as you said, is a graduate student in biopsych in my lab. She's in her, I forgot, like 4th year or so. So she's possibly away a good year or so from getting her PhD. And she has worked, as you all know, from her major publication last year on striatal cholinergic interneurons, and published last year a major paper showing that silencing those striatal interneurons is sufficient to actually reproduce the high fall rate we have seen earlier in the straightforward DL-RAD model of these Parkinsonian fallers. Since then, she has, her research has focused now, as you will hear today, on cortico-striatal information transfer in the context of complex movement control. And what will complicate a little bit her talk, I suspect, so you will have to pay attention to this, is that she's actually integrating into this research the animals that we have learned about a lot from our addiction research, which are these sign and goal trackers, because the sign trackers in particular serve as a natural animal model, if you like, for the relatively poor cholinergic attentional control capacities that, of course, we see at work in a much more severe degree in our models of Parkinsonian fallers and obviously in the patients. But as you will see, integrating those animals into research that is governed by the question, what happens in situations where you are using cues to guide your movements? What is the role of those corticostriatal projections in transferring movement cue information into the striatum? And how does that differ between animals that have a pretty good top-down attentional control versus animals that have, as a trait, relatively poor attentional control? And so with that, I'll pass this on to Cassie, and the title of her talk is Moving on Cue, Corticostriatal Transfer of Movement Cues. Cassie.
Okay. Yeah, let me just interrupt briefly. So I think with these kind of presentations, it's probably best if people hold their questions until the end or put them in the chat. Cassie, please go ahead.
Right, thank you. And I will try to monitor the chat as best as I can.
Yeah.
So I'm just going to go ahead and jump right in. Thank you, everyone, for being in attendance, first of all, right before I get in. Okay. As stated in the title, Moving on Cue, Corticostriatal Transfer of Movement Cues, today I'll be discussing how environmental cues engage and are encoded by our attentional processes to guide movement. So looking at this top left image, we can see an example of everyday behavior walking down the stairs. This may be perceived as simplistic or even leisurely behavior. However, in this example alone, there are numerous exteroceptive and interoceptive cues that, if not attended to, may precipitate a stepping error. For example, a particularly salient cue, such as a chime indicating a text message, may throw off the attention of this particular person, throwing them off or disrupting their pre-programmed movement sequence as they were making their next step down. Interoceptive cues such as proprioception, knowing where their foot is in space, or external cues such as the steepness of the steps, how close their friends is, they must be evaluated together to ensure corrective action like rebalancing their weight to prevent a movement error that could lead to a dangerous outcome such as a fall. So even a seemingly simple task we engage in daily, such as walking or walking down the stairs, requires intervention from our attentional system. And here I'll be discussing that neuronal circuitry that's implicated in this attentional regulation of cue-guided movement, as evidenced by experiments that we've conducted with our primary subjects shown here on the bottom right. In this image, their movement is guided primarily by interoreceptives or internal cues. As you can see that these rats will utilize their tails for balancing as they move across dynamic surfaces. In the experiments from our lab, however, the rats must utilize external cues that engages the circuitry that I'll be detailing on the next slide. So here is a graphical representation of the neuronal circuit that I'll be discussing here for complex movement control. And I'll be reviewing this in a simplified manner. So starting at the basal forebrain, key regions such as the nucleus basalis, a minor, the substantia numinata, and the horizontal nucleus of the diagonal band. The cholinergic projections to the cortex and cholinergic signaling within the cortex are necessary for detecting task and movement cues, such as the ones listed here as indicating dynamic surfaces, slips, and imbalance. The process of and the results of this such processing in the cortex is then transferred to the striatum via glutamatergic terminals, where it is then integrated with the midbrain dopaminergic signaling that's usually mediating action selection. Most of my talk here will be focusing on this specific component, the cortical-striatal transfer of task and movement cues. But first, I wanted to give some context in terms of the cortical cholinergic signaling in the context of attentional task that is the mainstay of our lab, the sustained attention task. So the sustained attention task requires an animal to report by pressing a paired lever the presence or absence of a cue. In this case, in the graphs over here, a light stimulus. This graph showcases instances averaged where the animal detected a cue and correctly reported the presence of this cue when probed seconds later. These animals in this experiment were implanted with sensors that allow the experimenter to measure the concentration of extracellular choline. I'll be getting more into the specifics of this methodology later in the talk. So just to go over this briefly, on the y-axis, there is the concentration of choline in micromoles, and on the x-axis is time in seconds, with this 0.0 being cue presentation. So as you can see, the concentration of choline rapidly increases in the cue detected state right after cue presentation. However, if we were to look at this in instances where the cue is missed, we don't see this increase. Going further, if we then take out this cortical cholinergic component via cholinergic specific neurotoxin, as you can see detailed here, lesioning the brains of our experimental animals with this toxin shown on the bottom right. These dark lines shown on the top over here are cholinergic positive fibers, so you can see the depletion in our lesion animals. If we are to have this cortical cholinergic deafferentation, what do we see in terms of signaling? So this is a graph that's similar to the first two that we saw over up top. This is the concentration of choline. This is indicating when the cue is on. And then we have a red and blue line indicating the two different types of trials I showed previously, where our outcomes where we have one where the cue was detected versus when there was an instance when the cue was missed. And we don't see a difference in the signaling here. So all this to say that cortical cholinergic deafferentation attenuates cue evoked cholinergic signaling. And so we know that this is impacting, we take out the cortical cholinergic system, it can attenuate this signaling, but what are the behavioral and attentional consequences of this deafferentation? So as shown in the previous slide, these are the representative coronal sections from animals with the lesions in the prelimbic cortex. If we were to look at the behavior as shown on the right, on the y-axis, we have the percentage of signals that are correctly reported. On the x-axis, we have signal duration. So this light stimulus has different durations, with 500 milliseconds being the most salient to 25 milliseconds, so a pretty short duration. If we're able to compare our lesion animals here in red versus our controls, you can see that they display a lower detection rate. So from all this together, we can deduce that cortical cholinergic inputs are necessary for this sustained attention performance. And this is all good and well, but if we were to look at this failure of cue detection in the context of movement control, what do we propose that this would actually look like? So here, this is a modified version of a graphical representation where there is a failure to detect cues, such as the cues indicating dynamic surfaces that might lead to slips and imbalances. The slips and imbalances themselves that are not detected as well because of this failure to detect. We therefore have this information not being processed and subsequently leading to attenuated cortico-striatal transfer of tasks and movement cues. This, therefore, would subsequently lead to impaired movement selection and sequencing. So going forward in my dissertation, I'm trying to model this, and I propose to do this by capitalizing, as Martin stated, on an animal model of diminished attentional controls, specifically sign trackers. But to orient everyone, I wanted to give a little bit of background about sign trackers and goal trackers. And these animal models that I'm discussing were discovered through via Pavlovian conditioned approach, or PCA. You can see this representation here on the left, where animals, specifically our rats, are placed in a chamber where an illuminated lever extends in the chamber for 8 seconds. It then retracts and a banana pellet is delivered over here to the goal cup. As indicated by the name, this is a case of classical conditioning, or where the animal doesn't have to work for this reward. Any association that is occurring is because we are pairing these two things temporally close. And there's different phenotypes that emerged from this PCA training that was observed. So animals may bias their attention to this cue or the sign, the lever, that predicts the delivery of the pellet, and therefore are designated as sign trackers. And you can see over here, they're really focusing their attention on this sign. They've even showing some consumatory behavior towards it. However, there is a different phenotype. An animal may focus their attention on the goal itself, using the cue just as a predictor for the reward and approaching the goal cup, as shown here, and they are designated, therefore, as goal trackers. These animals are These animals are assigned a score or PCA index to indicate how extreme of a sign tracker they are, the absolute being a +1, or how extreme of a goal tracker they are, the absolute being a -1. We therefore also have this subset of animals called intermediates. They're falling in between these ranges, and they don't show a preference, or they have moderate responses to these two different phenotypes. From the attentional biases that are on display on PCA alone, we can already propose that sign trackers rely on this bottom-up or sign-driven attention to guide their behavior, whereas goal trackers are showing displays of exerting top-down goal-driven attentional control, as they're using this sign or cue, the lever, just to predict the reward delivery and focusing on that, their goal. Additionally, sign trackers, we've known from previous research, when their attentional system is tasked by the sustained attention task, or it's SAT, they display poor top-down attentional control. And that's what I'm detailing on this slide here. On this first graph, up on the top left, the y-axis is the percentage of signals correctly reported. We have the y-axis, which is that signal duration, as I showed you previously. Here we're comparing sign trackers and goal trackers. And you can see with the sign tracker in green, the goal tracker in red, that sign trackers are displaying decreased rates of detecting cues compared to the goal trackers. Further, if we then plot their overall SAT score on the y-axis here, and we plot this against PCA index, which I mentioned previously, where the more extreme of a sign tracker would be closer to +1, more extreme goal trackers would be -1. So if we plot these against each other, we can see that higher degrees of sign tracking is negatively correlated with their SAT performance. The more of a sign tracker they are, the worse they're doing on this task. We can then also look at different components of attention where we can see how well they stay on task or their performance over time. Which is what we're showing over here on this graph. So we plot over here on the y-axis yet again. Hold on. How are they on? Okay, let me finish up this graph, and then I'm going to answer Cindy's question just so I can give a little bit of context for that question as well. Okay, so on this, oh So over here, if we're to look on the y-axis, which is SAT score, and this is plotted over time, which is on the x-axis in minutes, we can look at these representative sign trackers and goal trackers. As indicated here, the red is our goal tracker, the green is our sign tracker. And in this specific graph, good performance, favorable performance would be above chance. How we define poor performance is below chance performing. So we can see with the goal tracker that is in red here, that there is some variation in the performance, but overall, they're primarily staying in this good performance range. If we look at the sign tracker, however, there is a great degree of unstable performance that even worsens near the end of the task, showcasing a case of low vigilance to the task. So just some of my points here, and then I'll jump back to answering Cindy's question. We see that sign trackers are showing a lower detection rate. It's correlated with the degree of sign tracking, where the more of a sign tracker you are, the worse you are doing on SAT. And there is unstable performance over time. I'm going to pop up Cindy's question really quick and repeat what she's saying. So the question is, how are they on false alarms? Is this a
Let me also add emissions to that too.
Oh, add in omissions? Okay.
Yeah.
Is this a hits issue or an so how are they on false alarms and omissions? Is this a hits issue or an overall task issue or both? So I didn't get to too many specifics of sustained attention to tasks, but I previously mentioned that this is a task where the animals have to report this absence or the presence of a cue. So I'll just say for my case here that the animals, when they go through these tasks, they're in the chamber and this cue light comes on, like I said, with varying signal durations. After the signal light comes on, there's a brief pause and the levers extend. So they're going to be presented two levers. And the way that the animals are reporting the presence and absence of this is by pressing the correct paired lever. So for this example, we'll say that when the light comes on, it comes back, it turns off, and then the levers extend, they have to press the left lever. For them, that's how they indicate, yes, there was a signal. There's also these trials where there's the absence of cue, a non-signal trial, where the light isn't on, the levers extend, and now the animals have to report on the right lever by pressing it. There is not a signal. All this was just to say that there are instances, as Cindy's asking about false alarms, where the animals had a non-signal trial, but they still reported that there was a signal, a false alarm. So in terms of that, and I think the other part was omissions. In terms of omissions, I'll start with that. Omissions just meaning that they're not doing anything. They didn't hit either lever. So that would be just a signal that maybe they're not engaged with the task, they're distracted, so on and so forth. They're just not engaging in the task. And overall emissions, they haven't seen any differences with sign trackers and goal trackers once they get to this point of extended training. But in terms of false alarms, oh, sorry. Yes, sorry. Someone asked me something. So in terms of how this is calculated within the score over here on the sustained attention task score, right over here in this top graph, I'm only talking about hits particularly when they correctly responded that there was a signal trial. Over on the SAT score over here, that is for the SAT score, we actually are integrating both false alarms, them correctly saying that there wasn't a signal, and also omissions are all together reported in here. But in terms of for sign trackers, it really seems what's driving this particular this correlation really has to do with the hits, them being not being able to correctly report the signal. Therefore, in terms of this type of correlation, this is really due to their lower detection rate, not necessarily that they are emitting more, so to speak. Was that, did that address your question, Cindy?
Yeah, I mean, I'm coming at this as the cognitive psychologist and trying to nail you down. And as I'm sure you know, Martin and I have had similar conversations before about, so what specifically are the cognitive operations being supported by this. And I'll come back to that later on in your discussion.
Okay, yeah, no
Thanks.
problem. Okay. Okay. So as showcased just in this slide, sign trackers are displaying this poor or dampened top-down attentional control. But to get more back into the neurobiological bases, what would be these neurological bases for this diminished control? Okay, sorry, my slides were freezing. So in this particular experiment, there was a microdialysis probe that was implanted into the prelimbic cortex of sign trackers and goal trackers performing the sustained attention task. If we look over in this graph, at baseline, these animals do not show differences in the basal levels of extracellular acetylcholine. So this y-axis is % change from baseline. And if we are to look at the task during the task period, where that's indicated by this task onset and task offset markers here, when we're to look at this, there's increases in acetylcholine release during the task that is significantly higher in goal trackers when we compare them to sign trackers. These sign trackers do not seem to be elevating to the same degree of goal trackers. This then begs the question if this failure to generate cholinergic signaling that supports cue detection is what's mediating this poor top-down control in sign trackers. Are they failing to generate the signaling and therefore not detecting cues? And an important mechanism that I'm not going to be discussing in great detail, but evidence within the lab, is that we know that sign trackers have this dampened cholinergic signaling system, and it's mediated by their high affinity choline transporters failing to support further increases in cholinergic activity. And an important case to note is that the high affinity choline transporter is the rate limiting step of acetylcholine synthesis. So it's by this mechanism that they are failing to further increase any acetylcholine to meet the demands of a taxing task. So like I said, I'm not going to be getting into too much details of that, but we can talk about that later on during Q&A. But going forward from this, do these animals with the diminished top-down attentional control, the sign trackers, and attenuated cholinergic signaling, are they also showing impairments in movement control? And the short answer is yes. The paper over here that I'm detailing from is called Addiction Vulnerability Trait Sign Tracking Impacts Complex Movement Control. So in the video I'll be showing you over here on the right, I'll be showing you the different challenging and varying beams that animals have to traverse. And these types of beams have allowed the lab to identify distinct strategies that's goal trackers employ, sign trackers not so much. And not in this video, but it is an important thing that I wanted to mention is the straight rod beam that we employ. And there's different conditions that we have this where the straight rod that they traverse can be stationary or it can rotate in a clockwise or counterclockwise fashion. The counterclockwise fashion is the one that they're more familiar with and trained on, where you can then further probe them by rotating this now in a clockwise fashion. So specifically from that condition, they found that sign trackers fall and slip more often with goal trackers while they're traversing that straight rod when it's in an unfamiliar direction. And this can also be shown when they're tasked by a secondary task or a distractor. So this will make more sense now that I'm going to be showing this zigzag beam over here. And just to preface this, the video is going to show segments of two stationary conditions of the zigzag beam and another condition where this beam is consistently rotating. And you'll be able to see how the animals go about traversing this dynamic surface. Okay. So over here, the animals are entering this beam as when this goal is the goalpost is lowered. They don't want to stay out in this open space, so they readily are crossing over to this other post right here. And some important things to note about how they're doing this is you can see they try to go for flat surfaces, and they really also time their movements as well using those tail flicks that I mentioned previously to gain their balance again. So this is another condition where you can rotate the zigzag beam And now it has these steep inclines and declines, making it an even more difficult dynamic surface. Okay. And lastly, this is the very dynamic condition where it's consistently rotating. And you can see more of those different types of strategies that they're employing. And just to get a little bit into the details of that, of what the strategies that they employ and also how this differs between the phenotypes, the results from the zigzag beam showcase that goal trackers avoid entry into the beam angles when they're at a very risky or difficult position. So when they're at those steep inclines, the goal trackers are actually going to wait out until the beam rotates in a favorable position. So they not only are they employing this type of strategy, they also have to be able to balance while they're just slowly shifting themselves until it's in a favorable position. And that strategy alone is indicating that they're using superior top-down control. However, the sign trackers, they enter very rapidly and quickly and at risky spots. So where those angles of the zigzag beam were nearly vertical and very steep. As expected from that, sign trackers are falling more often while they're traversing this rotating zigzag beam compared to goal trackers. And one thing I will note that's just kind of an interesting thing to note is that this rotating, their falls in this rotating zigzag beam they didn't necessarily also have slips, and we can get a little bit into why that might be important further on, but I just wanted to basically tease you with that. But taken together, the findings that sign trackers display attenuated cholinergic signaling, diminished top-down attentional control that therefore disrupts movement control in dynamic environments. I wanted for my experiments to capitalize on these differences and perform electrochemical recordings from these animals while they were undergoing a cue-triggered motor task. Okay, there we go. So looking back at this circuit, and what I'll be primarily focusing on is not necessarily this detection side of things where we have this cholinergic signaling and also cholinergic afferents over into the prefrontal cortex. I'm looking at the further downstream effects of this in terms of the cortico-striatal transfer of task and movement cues. That will be the primary focus of my experiments going forward and in my dissertation. But my hypotheses were informed by the circuit displayed below. So if we have the detection of cue needing these corticocholinergic signaling, and therefore that, the cortex is therefore sending glutamatergic terminals to the striatum, I am therefore looking at terms of glutamatergic signaling within the dorsal medial striatum for my projects. So the hypotheses that are derived from this is that if I'm doing this recording is over in the striatum, glutamatergic signaling in the striatum is going to be encoding cue presentation in a cue-triggered motor task, reflecting that upstream corticocholinergic signaling. Additionally, if I'm going to be looking at these different phenotypes with sign trackers showing this diminished attentional control, sign trackers therefore will also show deficient or aberrant glutamatergic signaling compared to goal trackers. This would be then for secondary to their attenuated corticocholinergic capacity. And the approach that I'm taking to do this, like I stated, is to do amperometric or electrochemical recordings in the dorsal medial striatum of these animals awake and behaving in this cue-triggered turning task. So just to orient everyone to the cue-triggered turning task, this task specifically was inspired by clinical observations in Parkinson's patients. As Marvin mentioned, specifically Parkinson's patients fallers, says there is a subset of patients that fall and those who don't. So we're specifically interested in Parkinson's fallers. And Parkinson's fallers also show difficulty in turning. They generally need more time to turn. And another observation that was really important is that failures in this cue evoked turning precipitated or predicted falls. So we're going off of those clinical observations when we are going about constructing this paradigm specifically. And in this paradigm, the animals are placed on a treadmill belt that has a Faraday cage mounted on top. That's important as I get later on talking about our subsequent recordings to shield any electrostatic interference throughout the room. And that can be given off by outlets or even static on our clothes. So it's very important that we have this Faraday cage. And briefly to describe what the animals actually do on this is they are on this treadmill belt. It starts, the animals walk at a brisk rate until there is a cue presentation for about 2 seconds. A second later, there is this brief treadmill pause, and the treadmill belt will restart either in the same direction or the reverse direction. So if the animals are on this and the belt is restarted on the reverse position, the animal must turn as opposed to them briefly stopping if the treadmill belt is restarted in the same direction. So the rats here have to learn to associate the given turn cue, as we call it, with the reverse of the treadmill belt. On the other hand, they have to learn to associate the stop cue with the brief treadmill belt pause and restart in the same direction. So going forward from here, the types of recordings that I'm conducting from these animals is amperometric recordings via microelectrode assembly biosensors. These biosensors are chronically implanted into the dorsal medial striatum, also known as the projection area of the prelimbic cortex, making the case of why we're recording from dorsal medial striatum. These sensors have glutamate oxidase bound to the surface. With the application of a constant fixed potential, breaks down any glutamate that contacts these sites as shown here. This breaks down glutamate to hydrogen peroxide, which is subsequently oxidized, and the change in current due to these released electrons can then be measured. We then do this post-hoc analyses to convert this to the concentration of extracellular glutamate. And I use this method and recorded from sign trackers and goal trackers while they were awake and deforming on the CTTT. And just to orient everyone to this figure, because I know there's a lot going on, I'm going to break it down. So over here on these blue arrows are indicating movement direction. So this is just a way so we can see that since they're facing each other, this is indicating that there's a reversal. So these are our turn trials. These markers over here on the left are denoting our measurements. So about this much would be a 1 second. And over here, this would be equivalent to about 5 micromoles of glutamate. Oh, and then lastly, before I get a little bit into this, is in this red area with these dashed lines. This is showing Q on and Q off. So this is the Q presentation area of the graph. So as expected, detected turn cues elicited glutamatergic spikes when, as opposed to these missed cues shown in the lighter blue. And this we were able to see in goal trackers. So this was aligned with what we were predicting. However, when you then look at the average data in sign trackers, it was not what we expected. We were expecting, just to preface, what we were expecting was attenuated glutamatergic signaling, and this is the exact opposite of our hypothesis, where all turn cues elicited this glutamatergic spike regardless of trial outcomes, regardless if they had turned or not correctly. And so in this, the glutamatergic signaling in sign trackers, it doesn't seem to discriminate between Q trials or missed trials. And I just want to preface this by saying that this is average data that has been useful when we're looking at this macroscopic view of how these phenotypes differ in their signaling. But going across in terms of how informative this is, it could mask different characteristics in these signaling properties that we could therefore use to do things such as trying to predict trial outcome. So as I'm saying, this is very informative to just look at the overall differences in side trackers and goal trackers, but it's not very informative because we are averaging across animals and across multiple trials. So moving on, what we'll be doing in terms of analysis is looking at individual raw traces. So just to get an idea of what that looks like, we have representative traces from a goal tracker and sign tracker shown here. And you can already appreciate the differences that we are seeing between these phenotypes, but also when we aren't averaging across animals. And we'll be utilizing these differences that you can see here by defining different characteristics of the signaling properties and then extracting it from these raw traces. So this different characteristics that we have characterized here so far is one, number of peaks shown here with goal trackers on the left column, sign trackers on the right column. We do already see that sign trackers seem to present more spikes. The time to peak in seconds seems to be a bit faster in goal trackers. Time between peaks, we don't have that measure in goal trackers at the moment, as they're only presenting that one peak. Max amplitude in micromole seems to be relatively similar. The time to max amplitude, however, is faster in goal trackers. And therefore, when we look at things such as slope, it's greater in goal trackers. As we said, they are peaking faster and reaching that first peak faster. So therefore, you would expect that their slopes are faster. And these different characteristics are, one, it's interesting to see. Two, what we therefore can use these characteristics as are potentially as classifiers or predictors. We can go back and look at the behavior and see if these certain signaling characteristics, if they predict whether or not they have a successful turn, what is the trial outcome? And that's what the types of measures that we hope to, analyses we try to do, hope going forward. And one second. Just to wrap up a bit of what I've shown you so far is medial dorsal striatal glutamate signaling encodes cues leading to successful turns, but that's only in goal trackers. And it begs the questions if the absence of this turn cue evoked glutamatergic signaling is causing missed turns. It's kind of a chicken and egg situation here. And the ways that we're going about trying to address this question is different mechanisms, like I stated before, analyzing glutamatergic spikes as prospective determinants of determinant of turns of the trial outcome. The other methods that we can employ that I'll be going to talk about in the future directions is demonstrating the behavioral impact of glutamatergic silencing and also of cholinergic losses at large. If we then look at sign trackers, however, glutamatergic spikes did not predict trial outcome, and yet their acute turning performance was not affected. They're still performing at the same level of goal trackers when we look at their performance on the cue triggered turning task. And this indicates that there is some alternative task cue sources, perhaps via striatal afferents from the PPN, that still allows them to perform at the same level of goal trackers. Further, this non-discriminative action of this bidirectional glutamatergic interneuron interactions in the striatum supports striatal output generation. However, as I stated before, the source of this well-established and habitual cue control remains unknown. So going forward to how we might parse out the role and impact of glutamatergic signaling in goal trackers, I have a few directions that I'll be implementing. So one of them is to look at the impact of corticocholinergic deafferentation as shown in the previous experiments, therefore leading to this failure in detection of cues and potentially the cortico-striatal transfer of task and movement cues. The other method and direction I'll be taking this in is to use a dual vector approach to silence the cortico-striatal pathway. So in this, I'll be infusing a decree-dependent inhibitory dread and virus into the prelimbic area of these animals. On the other end, I'll have a retrograde Cree virus that will be infused into the dorsal medial striatum. So when this retrogradely transfects, it meets up with this virus, we can therefore have selective and specific silencing of the corticostriatal pathway when we have our designer drug on board. So if we're looking at these two approaches, we expect that the manipulations would have the same impact on goal trackers, that they will show decreased cue-triggered turning and attenuated glutamatergic signaling, but this manipulation should only impact goal trackers, but not sign trackers based off our evidence, so to speak, now. And before I get too much into questions, I just wanted to give a few acknowledgements. In particular, my dissertation committee with Natalie Tronson, Omar Ahmed, Shirley Flagel, Mark Bradshaw, who was our former fabrication specialist, who also made these treadmills that I can end up utilizing for this. By our biopsychology graduate students, my fellow graduate students, in particular, Kit Silico, Ileana Morales, and Chris Turner, who have been endless supplies of support. All the rats, obviously, funding sources, in particular, of course, Udall, that will allow me to continue these studies. And I would be remiss if I don't acknowledge my lab, Barton Sarter, my primary mentor, and my colleagues within the lab, fellow graduate students, and our postdoc, Aaron Katinsky, who's done a lot of that more MCT related, the beams related work that have derived a lot of what I've continued to study. My lead undergrad RAs who've helped me immensely, and former members that I just wanted to make a quick note about. Kira Phillips, who was a senior graduate student when I entered the lab, who was a great mentor as well and traversing the lab. And my former RAs, Annie, Ellie, and Stephen, who were pivotal to me even collecting this data, running all the animals, helping me collect the data, and even performing some surgeries. So with that, I'll thank you all for your attention, and we'll take any questions.
Excellent. Okay. So, okay. Yeah. That was a really nice talk. Thank you. Question. So you went over your next steps pretty well, but you mentioned that you think the decision in turning that might be somewhat farther downstream. So what level do you think that is?
In terms of the sign trackers?
Yeah.
Okay. Let me go. Well, actually, I don't even need to go back there. So one of the possible ideas we have where that decision might be happening. Well, first off, just to state that just based on their attentional biases and how the classical research has been for sign trackers, we believe that they're using more bottom-up types of methods, so more subcortical areas. And one of the ones that I mentioned very briefly in the conclusions was potentially acronyms from the PPN.
Yeah.
And one of the leading reasons we believe that it might be the PPN is when you look at the signaling properties within there, these cells are broadly responsive to any task event, which would be in line with what we were seeing with the glutamatergic signaling within sign trackers. So that's one possible mechanism, and that would be more dopaminergic related mediation, which we know is important for sign tracking, more in the motivational side of things, which I don't fully study in the lab, but it would fall in line with that type of research.
Okay.
Hi, Cassie, great talk. I have a number of questions, but first I just want to say I'm really glad that Roger asked the question that he did, because I was originally going to try to make the connection to some of our ERP data in OCD patients, because they show what looks to be a continuous firing, but if anything, they're probably too high on cholinergic. So I think you have a great example there of how multiple roads can lead to Rome. And I think you're probably on the right track with that. So I have a couple of questions, and hopefully this doesn't feel too much like your dissertation. And if it starts to and we need to take it offline, then feel free to say that. So actually, let me start with the one that is probably most closely related to this, and that relates to the beginning of your talk. So one thing that you glossed over a little bit is that the cholinergic response, at least as I understand it, and as Matt Howe understands it, in the DSAT is sequence dependent. And so there's been different possible explanations for that. Given that you think that the glutamatergic response is sort of the passing of the baton, I'm curious as to whether that's even more sequence dependent, because that could sort of, again, kind of help us figure out which aspect, basically how each of these is contributing to different parts of this causal chain.
Yeah, no, I think that's a great question. I definitely did gloss over it. I even glossed over a little bit of what cue detection necessarily means. But just to help everyone get a reference here, we know that from the past research that cholinergic transient activity is particularly important for when there is a needed attentional shift, when animals are going from this cue monitoring state, they're like, yes, there's a cue. And then they have to actually enact in terms of a cue directed behavior. In terms of sequencing, first I'm just going to briefly mention, we are going to be looking at that in terms of the analyses. So as I said, the average data tends to mask a lot of things. So for the individual traces, we are going to be ranking those in sequences to see if we do see any of those sequential effects. I'm just going to go based off what I've seen within my research. I haven't seen this.
To see it for your task, actually, because I think the shift in behavior is so big as opposed to in SAT.
Yes, that's part of it. The other part is, I mean, we're talking about within SAT itself, we already see those differences with sign trackers and goal trackers. I'm not seeing it at the level of my task, which there's a few different potential reasons. One, it could be that the circuitry itself is so highly redundant for sign trackers that that's how they're able to perform. The other part of it is it might not be taxing enough for my task, and that they can still, not necessarily have to rely as much on the attentional system as they do for something like SAT, where you have it going on for 40 minutes. They can get probably pretty boring. They have to report different types of trials, whereas mine's just mostly just like turn or no turn. No, thanks so much for coming. So yes, from the preliminary data, when I looked at the data, I haven't seen anything in terms of sequences, but like I said, we haven't really looked at the individual traces as closely, so I'll have to keep an eye out. But as you're also saying, I'm not sure if I'm going to really see anything in my task.
Yeah, I would be actually surprised if you saw it for your task, just because, like I said, the shift, if you think of it as a task rule shift, that's a pretty big shift regardless. I do have a question sort of linking, so this will be my last one, I promise. Also linking back, since you had made the connection to Aaron's work with the rod rotating and the goal trackers. So, part of the way that you describe the goal trackers there, in terms of sort of like their planning behaviors, one could think of as being really more top-down proactive planning, and reasonable people can disagree as to how much that is, cue detection is all and everything versus whether there may also be some other cognitive operations out there. So one thing I'm curious about is you have enough dynamic range, it looks like, in your goal trackers, even on your task. Would you expect, so let's say if you gave them like a colored light before the treadmill starts moving, and if the colored light is red, like there's an 80% chance that they're going to have to turn. And if it's green, there's only a 20% chance that they're going to have to turn. Would you expect to see that they're going to be better at making the turns after the red light, actually indicating that they are doing some of that plan ahead proactive control?
Okay, just to make sure I understand your suggested paradigm, this light, the red light versus the yellow light, would those be on during the same session, or is just one condition?
Sorry, I muted myself for a second there. So sorry, I probably shouldn't have chosen red light versus yellow light, since that's what you're using to cue the actual turn versus stop. Basically what I'm saying is, so if before the trial even starts.
Oh, before it starts, okay.
Right, there's a cue that lets them know. So this is like basically like a Posner pre-cueing. paradigm, but adapted for your task, right? So there's something that comes on that says like, okay, on this trial, there's an 80% chance that I'm going to tell you to turn, as opposed to on this trial, there's a 20% chance that I'm going to tell you to turn. Are they better at, and so I would only expect this for the goal trackers. But if they really are showing proactive control in that rod task, and this is a generalizable phenomenon, one might expect that then they would be able to do this. But if it's really just about cue detection, that might not make such a big difference.
Yeah, no, that's an interesting question. I don't want to necessarily take the easy way out or anything like that. One thing I would argue for, at least for the beams, one argument would be there for the rats in terms of what would be easier for them to employ is when we're talking about like what they preferentially are able to employ. So for the beams, they're utilizing this kind of strategy where they're timing it. They're one part of it that might be a little bit complicated with that is just that it is not necessarily that they're utilizing like this visual stimuli of the angles or anything. They might be utilizing more internal cues in that sense, and then just happen to learn this as they're going throughout to start to time their movements. And there is actually some data that suggests a little bit of this in the sense of like when they're going through the transverse beam, the zigzag beam, when they're first learning this, they do have a higher presentation of falls, both sign trackers and goal trackers, but the goal trackers learn as the trials go on to start timing this, start to utilize this strategy, and then especially once we start getting that rotation, this consistent rotation. So one of my arguments would be for that, just in terms of what information they're utilizing. For mine, they would have to be utilizing just that visual representation, which I don't think they necessarily would be the best at, considering the rats, especially the rats I'm using, since they're sprags, and the discrimination might be a little bit harder for them. But if we're just going to go on the pure cognitive side of things, that it's just that they're actually able to utilize that cue to have this prediction, or utilizing that cue that has a prediction of this, like it's 80% chance versus whatever. I'm trying to think of it in the context of my task. I know the previous student before me, Kira, had a little bit of differences of the rewards. Like there's an 80% chance of getting this reward versus the other. And there was differences there. I'm trying to think in terms of my task though. I'd imagine they would struggle to use that. As you stated before, yes, I don't think the sign trackers could do this at all. Even on my task alone, when they're first training up, even though they take about approximately the same amount of time to train up to the criterion that we have for them to get reported from, we do still see instances where the sign trackers do get a little bit distracted. They rear, they'll start looking at the lights where that we already have, that's predicted. So we have two cues. One can be a light versus the tone, but they warrant to both once they have these, and they do get a little bit distracted. They train up fine, but yes, I'd imagine the sign trackers can't do this at all. The goal trackers, I'm honestly not really sure whether or not. I would say that they probably would struggle. I'm not sure if they'd be able to use this other like priming cue to predict whether or not their turns. And also the case of their motivation to do so, I'm not sure if they would be motivated enough to actually even utilize that. They might just be fine with being like, I'm 80% correct. I'm not going to even employ that priming. I think that might be another confounding variable there, is that they might not even be motivated enough to use that cue and to learn about it.
Okay, so actually, I think we have one last.
Can I take a minute, Roger? Can I take a minute
please.
to come back to your question, which is the question. Where do sign trackers make this decision, right? Since the cortico-striatal input is not differentiating between trial outcome, you wonder where and how they're doing it. And the answer is they're doing it within the same circuitry, because look, it is a habitual response, right? These guys are well trained to do this. It's like you approaching a traffic light in the night and you know you have to stop, right? You can do this top down if you want to. If you are so inclined, you're thinking about, okay, I have to stop because I need to avoid an accident. And if you are driving home and your cortex happens to be completely knocked out, you will still do the stop because the striatum gets redundant information, specifically from the PPN, that knows pretty much about every task, event, and cue, as we know from neurophysiological recordings, right? And so it's not like you're knocking out the downstream decisional circuitry in the striatum, but you're flipping from a top-down version of
to a bottom-up
decision to an habitual decision, right? And so what's happening in sign trackers is basically the top-down influence to make this decision is almost removed or not existent, right? The surprise is, okay, why is it then that the glutamatergic terminals are so active even during missed turns? And I suspect this is simply because all differential regulation downstream and interactions with interneurons is basically abolished, and maybe just dopamine is now driving that input single-handedly to support the habitual response. But the striatum has that information about the cue, now please turn, or now please stop, right? You just now do this decision, basically completely devoid of any top-down response. I mean, it's literally, that's the best example I can come up with spontaneously. It's literally like driving home, being terribly drunk, the cortex is knocked out, you'll still probably manage to do a stop at an intersection, but you're not doing it top down. It's an habitual, quote by quote reflexive.
And you try this every,
I'm thinking about this.
And you do this every weekend, I'm sure, right? So no, actually, we have one last question, which I think is a very good question from Young-Soo Kim. Any speculation on the role of acetylcholine different cue-induced glutamate change patterns? I guess I would also add to that is there a thought that maybe those different goal versus sign track and glutamate signals have somewhat different sources, different afferents, perhaps?
Yes. So,
yes. Okay.
Yes, that's it. No. Yes, part of it is, as Martin had stated, that this is highly redundant circuitries. I mean, it's the basal ganglia, so pretty much the whole brain. They both have this integration. It might just be that there's predominantly this source that is the source for the goal trackers, and then this might be a predominant source for sign trackers. And they both still have these different brain regions involved, still have the same circuitry. It's just predominantly they're getting the signal from this certain source. So I would say in the speculation of the role of acetylcholine on different, I would say in terms of the way I guess I would say what area you're specifically talking about. If we're talking about the role of corticocholinergic acetylcholine, then we would say that there would be a role of the corticocholinergic choline on different cue-induced glutamatergic changes, specifically for goal trackers, but not for sign trackers, if that's what we are trying to implicate with our studies, in the sense that I'm not sure if necessarily I'm going to be saying that too much with what I'm looking at, but the idea here is that they are utilizing this information that's being forwarded from the cortex via cholinergic signaling here. So, and one thing I'm not going to get to talk about too much here is we do see some differentiation with turns versus stops in goal trackers. We see this signaling events, this signaling with glutamate specifically only in our turns and our successful turns for goal trackers. We're not seeing this with stops. It seems to fail to evoke any amplitudes that would suggest that they're actually needing this integration to perform these stops. So then it comes down to the question, like, what's so different with a stop versus a turn? The turn, I'm sure I'm speaking to the choir. It requires the integration of lots of, not only just that external cue to know when to turn, but also the internal cue and learning, and also the cues to rebalance, shifting your weight, finding your sequences in terms of limb placements and everything. So the actual requirements of a turn is a lot more heavily involved than our stops. So at least within the role of acetylcholine there, we would suggest that the enrollment would be a lot higher for our turns, specifically in goal trackers. There might be roles of acetylcholine, obviously, in sign trackers. The main reason I'm saying it just might depend like what we're talking about in terms of the neuronal areas, because we would be saying, oh no, acetylcholine doesn't have a role, where then we're disregarding PPM. We're also disregarding cholinergic interneurons within the striatum. So it really, I think it's gonna be very case specific, dependent on the neuronal region. And I won't get too much further into that because we're out of time and that would take a little while.
Okay, well, thanks very much. And actually, I'd encourage anybody to contact Cassie and Martin with further questions about, about this really interesting set of results. Just a reminder, we will have another meeting next month. I think, Nancy, I think we have a guest speaker. Is that correct?
Yes, Michael Vessia.
Michael Vessia from
Kinesiology.
kinesiology is going to talk next month. So, please come back. And again, thanks, Cassie, for an excellent talk and excellent discussion. So,
Thank you.